Here we're going to continue our discussion on fatty acid oxidation, and we're going to talk about other types of oxidation besides beta oxidation. So here we're just going to talk about the role of the endoplasmic reticulum and the peroxisome especially in fatty acid oxidation. So we talked about our saturated fatty acids, and we talked about how they were oxidized when they entered the matrix of the mitochondria. Now, if you have an unsaturated fatty acid which had double bonds, it's going to require additional reactions. We're not going to go over that. It basically is going to move your double bond around so that you can continue beta oxidation. We are going to discuss odd-numbered fatty acid because it's important and it could be potentially associated with diseases. We'll discuss very briefly medium chain and small chain fatty acid. We'll discuss very long chain fatty acid. It sort of is similar to odd-numbered fatty acid, a little bit different. And then we have, of course, our branch chain fatty acid. Very long chain fatty acid and branch chain fatty acid are both going to have to go through the peroxisome first before they continue on in the matrix of the mitochondria. Now, as we discussed, there's chain specificity in our enzymes. So we have many different enzymes, and they're all going to be specific for a certain chain length of our fatty acids. And in general, your long chain fatty acids are going to get transferred from enzymes that act on longer chains rather than those that act on shorter chains. So medium chain and short chain fatty acyl CoAs cannot work on long chain fatty acids, for example. So your medium and short chain fatty acids are going to come in through your diet, but they can also come in through your peroxisomes, as we're going to talk about. All right, let's first start with odd chain fatty acids. So oxidation of odd chain fatty acids occurs in the mitochondria matrix. As long as it is not branched. So you have an odd chain fatty acid here, and we're going to go through beta oxidation just like we discussed with our even chain fatty acids. And at the end, though, you're going to get one acetyl-CoA, which can go off and go onto the TCA cycle. And we're going to get a 3-carbon, and this is going to be Propionyl Coa. And so Propionyl Coa has to be dealt with. Now, what happens with Propionyl Coa is you're going to have to go through a process that requires 3 additional enzymes. In addition, you're going to require some cofactors as well. So we have a Propionyl Coa carboxylase, which is our second enzyme that uses biotin. Remember, pyruvate carboxylase used biotin as well. And in the mitochondria matrix, again, Propionyl Coa carboxylase will use biotin. And so you're taking Propionyl Coa and you're generating methylmalonyl CoA. And now your methylmalonyl CoA is going to have an epimerase, and it's going to change it from the D form to the L form. And you're going to get L-methylmalonyl CoA. Now in this next step, you're going to require vitamin B12 as a coenzyme. So vitamin B12 is going to be here, and you're going to generate succinyl-CoA via a mutase reaction with methylmalonyl-CoA. Now, of course, we know succinyl-CoA can then enter the TCA cycle. Now, it's going to enter the TCA cycle at a site distant from where acetyl-CoA enters, so you're not going to get as much energy out of your Propionyl-CoA than you had with your acetyl-CoA. Okay, so now let's very briefly about medium chain fatty acids. Medium chain fatty acids are going to be more soluble in water. They're going to be liquid, as we discussed. If you're 8 or less, you're liquid basically at body temperature. So this is not going to be stored. So after a meal, if you do have medium chain fatty acids, they're going to be transported primarily to the liver. They can also go to the kidney. And then your liver is going to move them into the matrix, and the matrix is not going to use the carotene transport system. They have monocarboxylate transporters as well that can pick up these smaller chain fatty acids. And then you're going to get activated to acetyl-CoA through beta oxidations, and then you're going to generate energy. Now, this medium chain acetyl-CoA synthetase has a lot of specificities, so it's going to be able to form acetyl-CoAs with things like aspirin, benzoate, and then they're going to get conjugated to glycine and excreted. So this is a way to, again, add charges to a xenobiotic so that you can excrete it. Now, if you have an increase of medium chain fatty acid or small chain fatty acids, acyl glycines in the urine, and this is with acyl carnitine or with our dicarboxylic acids, basically you're not able to oxidize these medium chain or small chain. It can indicate a fatty acid disorder. And so if you have a medium chain dehydrogenase deficiency, you're going to get an octonyl glycine that's going to be in the urine, and this is a common deficiency is this MCAD deficiency. Now another alternative route is are listed here. We're going to talk about in the peroxisome, we can have beta oxidation as well. It's the same process as we talked about in the mitochondria matrix with one exception, and that's that first step. So the first step of very long chain fatty acids is you're going to generate hydrogen peroxide. And this is because this enzyme in the first step is an oxidase. And you're going to transfer those electrons to an FAD attached to that oxidase, but there is no electron transfer chain. There is no CoQ. And so basically what happens, those electrons go somewhere, and oxygen loves to pick up electrons. So oxygen is going to grab those electrons and become hydrogen peroxide. Now this will get quickly neutralized by catalase. However, you lose that energy potential in that first round of oxidation in the peroxisome. There's also peroxisome alpha oxidation, and this is where you have branched chain fatty acids. Now when you have a branched chain fatty acid, before you can even start peroxisomal beta oxidation, you have to do peroxisomal alpha oxidation. We'll talk about that. And then there's also microsomal W or omega oxidation. This is with our P450 enzymes. And this is very minor. And it only usually occurs to high levels if there is some sort of deficiency or your system is overwhelmed with too many fats. All right, so let's talk about our very long chain fatty acids. So our very long chain fatty acids are going to be picked up. You're going to have a synthetase that's going to activate your very long chain fatty acid, so it's activated. And then the very long chain fatty acid is going to cross the peroxisome membrane and enter the peroxisome. Now inside of the peroxisome, that first step is going to be by an oxidase, and this is going to generate hydrogen peroxide. And then after that step, you're going to have the hydratase, just like you have for beta oxidation. You're going to have a dehydrogenase, which is going to donate to NAD. And then you're going to have that same thiolase that's going to generate an acetyl-CoA. So you are going to be able to generate NADH and your acetyl-CoAs. Your peroxisome also has the carotene system that can take your CoA and convert it to carnitine, and then you can move your small chain fatty acids and medium chain fatty acids out of the peroxisome, and though you can actually go to the mitochondria and continue beta oxidation in the mitochondria. All right, so acetyl-CoA that's generated out each round of beta oxidation, as well as medium chain or small chain fatty acids, are all going to go to the mitochondria and complete oxidation. And so this is just showing them entering, and they undergo further oxidation. Now hydrogen peroxide that is made in the peroxisomes is going to be neutralized by catalase. So peroxisomes are going to be in almost every single cell, and that first fatty acyl-CoA oxidase is going to generate hydrogen peroxide because, again, those electrons have nowhere to go, so oxygen is going to swoop up and take them. Catalase is going to convert your hydrogen peroxide to water and oxygen. And one thing that's important is, of course, that your peroxisome enzymes stay within the peroxisomes and they don't get released into the cytosol. Now, another type of oxidation called alpha oxidation occurs in the peroxisome as well. So there is a phytanic acid, which is found in fish and some dairy products as well. And this is going to have a methyl group on your beta carbon. And the methyl group on your beta carbon is going to prevent beta oxidation. You're going to have to do alpha oxidation before you do beta oxidation in the peroxisome. So basically, we start with phytanic acid in our diet. So this comes from our diet. And then we're going to have an aldehyde dehydrogenase that is going to work on phytanic acid to make pristanic acid. Pristanic acid is in a format that now the normal enzymes in the peroxisome that do beta oxidation can now do beta oxidation. And the oxidation occurs same as very long chain fatty acids. Now, pristanic acid it's important because phytanic acid is in normal things in our diet, like milk and fish, for example. You have to undergo alpha oxidation first, so this is first, before you can do beta oxidation. And so this is going to be second plus. There are some defects in alpha oxidation. Some people do have defects, and this is going to lead to a disease, refusum disease, and this is a neurologic disorder because you're basically going to not be able to break down phytanic acid. And of course, a lot of babies, or most babies, have dairy in their diet, and so it's going to build up phytanic acid and be detrimental to brains and neural function. Now, another oxidation is omega. Omega oxidation is in the ER. Again, it's kind of like a backup system. You're going to have your P450 system making. So again, we're moving around electrons. These all use NADPH. And then you have the potential for reactive oxygen species. So the two P450s that do omega oxidation are CYP4A and CYP4F. And so this is going to occur primarily in livers and kidneys. And it's just basically you're going to take your fatty acid, you're going to make a fatty omega hydroxy acid, and then again, eventually you'll generate your acetyl-CoA from that. So in the peroxisome, you need to be able to break down very long chain fatty acids. So if you have any defect of proximal enzymes, you're going to have a buildup of very long chain fatty acid, which is not good for cells. You also have to do branch chain fatty acid oxidation in, not branch chain, you have to do your phytanic acid oxidation, your alpha oxidation in your peroxisome as well. Now, both of these are not feedback regulated. They are feed forward. So if the substrate is present, they're going to metabolize it. Now, omega oxidation, again, is going to occur in the endoplasmic reticulum as a backup system. Okay. So here we just talked about different ways to oxidize fatty acids beside beta oxidation in the mitochondria matrix. And so we learned that medium chain fatty acids and small chain fatty acids are not stored but get oxidized. So if that's part of your diet, they're going to get oxidized immediately. Your odd chain fatty acids and very long chain fatty acids both have to go through oxidize in the peroxisome first. Your peroxisome is going to do alpha oxidation. Peroxisomes are going to generate hydrogen dioxide during the first step of beta oxidation, and the ER does omega oxidation. Odd chain fatty acids, this is actually wrong, sorry. Odd chain fatty acids are going to generate Propionyl Coa. And this can become succinyl-CoA. And this can go to the TCA cycle. So this is mito matrix.